So in this chapter, we're going to revisit some of the nutritional requirements of microorganisms that we saw or we were introduced to in the last unit. So ultimately, the growth of an organism requires metabolism, right? And when we think of the word growth in multicellular organisms like ourselves, we think size. But in microbial growth, we mean population size, or the number of individuals. And so when we look for them in culture, we look for discrete colonies, which you recall we use the term CFU, or colony forming unit. This is actually how labs report the number or the amount of growth of a culture. All metabolic reactions require two things: a nutrient source, which of course is typically based on carbon, think carbohydrates, fats, proteins, things like that, and also an energy source to drive those metabolic reactions. So if you put these combinations together, if you took inorganic carbon dioxide, which is autotrophic, you would get those photosynthetic organisms that use light to drive those reactions. So if you put the auto and photo together, you basically get this big word, photoautotroph, which basically describes those organisms that are your typical photosynthetic organisms: plants, algae, even that small group of bacteria that can do photosynthesis. And so if you think of photosynthesis, most organisms that would come to mind would belong in this block. Now, if you look at a rare form of photosynthesis, those that use light to drive reactions, but they're not using inorganic carbon dioxide, they're using organic forms. And so this is a rare group of bacteria, which we would refer to as photoheterotrophs. Now, remember, hetero means other. So auto would be self-feeding. Hetero would mean other feeding, or feeding on other things. There's another small rare group called the chemoautotrophs. If you use inorganic carbon dioxide, but you use chemicals like sulfur to drive your reactions, then you would be in this group. Again, these are going to be rare types of bacteria. And virtually everything else on the planet lives here, what we call chemoheterotrophs, including ourselves, right? We use organic compounds like, again, sugars, fats, proteins, and we use chemical compounds such as ATP to drive our chemical reactions. So it's just a way of categorizing living things based on their nutritional needs. Now, your textbook also has this form of that same information. It's just laid out a little bit differently. So recall that in order to move things across membranes, we rely on transport. And so there are two general types of transport processes, which we call passive, which requires no energy. The thing that drives those types of transport is just having a concentration gradient of substances on one side or the other of a membrane. And so if it's simple diffusion, it just means those molecules are moving from a higher to lower concentration. If it's facilitated, typically this is where a membrane is involved. And so in order to get things past that membrane, typically it needs to be facilitated, or there has to be a place for it to cross. And so in this case, you would have things like channels or gates that specifically fit some substance that needs to cross over. If you need to move things against their concentration gradient, we would use active transport. And so this is where again, you need a protein passageway of some sort, but in this case, they're probably going to be referred to as pumps. And sometimes those pumps require ATP, or I should say most of the time they require ATP. Sometimes they can use something like GTP, but typically it's ATP. Either way, it requires energy to move things against their concentration gradient. And so we would call that active transport. When we think about the term osmosis, we're talking about the passive transport of water, but it's moving against the concentration gradient. So you may have heard the term that water chases salt, right? Well, that's basically defining osmosis. So this idea of osmotic pressure really determines where microorganisms can live. If you remember these words, solution is just a combination of solutes, or the dissolved substances, and the solvent, which is the liquid background of the solution. So if you have an isotonic solution, it means the concentration of solutes is equal on either side of that membrane. And so depending on where you live, if you're in isotonic solution, the water will move across that membrane. It's just going to move in the same volume or the same net volume. And so you won't get any change in the cell necessarily. However, if you're in a hypertonic environment, of course, the prefix hyper means more. When we're referring to solutions, we're talking about the condition of the solution outside of the membrane. Okay, so if it's hypertonic, it means there's more salt outside, less salt inside. And when I say salt, any solute, but salt tends to be the biggest driver when it comes to osmotic gradients. And so if you didn't have a cell wall, that water would leave the cell and the cell would crenate or shrink, right? If you have a cell wall, it would do the same, but the cell wall would maintain its integrity, its structure. It's when we get into hypotonic solutions that we really derive osmotic pressure gradients. And so hypotonic, meaning less solutes outside of the cell, means that the water is going to want to rush in. And so this is, again, why cell walls matter, because if you don't have a cell wall, that flexible cell membrane could burst. Organisms that have cell walls, like bacteria, fungi, algae, those organisms could take on water and not damage that cell membrane. And so this will determine where these microorganisms can live, right? If you are an obligate halophile, remember that prefix halo is referring to halogens, which is the if you look at the periodic chart, it's the next to last column on the right, and that would be things like chlorine, fluorine, iodine, and so that chlorine is the salt portion here. And so if you're an obligate halophile, you actually need an environment with a high concentration of salt. Some organisms are facultative halophiles, which means they can tolerate higher concentrations of salt. So Staphylococcus, for example, would be one of those organisms that would be facultative halophiles. They don't mind the salt on the skin, whereas organisms that are unable to tolerate that salt, it's the osmotic pressure that's going to damage them or kill them, so to speak. We talked about temperature ranges, and we met these terms earlier in Chapter 2 when we were talking about culturing conditions. So again, remember that there's always an optimal range for each species. And I realize in your textbook they call this psychrotolerant group. They also call it psychrotrophs, same group. And the hyperthermophiles, your text also calls them the extreme thermophiles. Either way, we're talking about this optimal range that they can survive within. Anything outside of that range, they won't be able to survive. We also have to consider oxygen requirements and gases in general, but usually oxygen and carbon dioxide are the most influential when it comes to growth. Oxygen in particular, because oxygen drives the electron transport chain that makes oxygen. We're going to see that electron transport chain when we get to Chapter 7. And the oxygen can also be quite toxic. And so because it's oxidizing, it's going to be something that can damage cell membranes. The fatty acids in cell membranes are very easily oxidized, and so we'll talk about that possibility. And so when we talk about organisms that can or can't survive in the presence of oxygen, you're going to have three groups. You're going to have those that can use it and detoxify it, and these are typically going to be the ones that rely on oxygen heavily, and so they've evolved to have ways to detoxify the oxygen that they produce themselves that might be toxic. You might have some that have no interaction with oxygen, meaning they can't use it and they can't detoxify it. So they're going to actually be controlled by oxygen because they can't tolerate it. And then you have some that don't actually use oxygen in their metabolic reactions, but they can survive in the presence of it because they have enzymes that can detoxify it. So keep in mind, most chemical reactions involve oxygen in some way. And so when those byproducts that are produced are produced, some of them are in these toxic forms. These are just some of the most common toxic oxygen forms, what we call a superoxide ion. This is basically an oxygen molecule with an extra electron tag to it, right? This is not the usual condition for oxygen. It should be just O2. You can have a hydroxyl radical where that oxygen and one hydrogen has retained an additional electron. Or you can have what's called singlet oxygen, which is, again, just on its own without any charge. And this actually is a very powerful, very reactive molecule that is actually one of the tools that phagocytes can use because it's a very powerful oxidizing agent. So we talked about phagocytes using lysozymes. They can also use singlet oxygen as one of the ways that they can destroy their enemies. And then we have hydrogen peroxide, which of course is also very toxic to cells and in such a way that it's actually used intentionally as a disinfectant and antisepsis, which we'll see in chapter 9, where we're going to try to control organisms in the environment. So because it's toxic, and those cells that are exposed to it have developed enzymes. So if you don't have an enzyme that can detoxify it, it's problematic. So these enzymes typically can deal with these oxygen forms in one of two ways, or in a sequence of two steps. And so this very common enzyme that's found in microorganisms is called superoxide dismutase, and very oftentimes you'll just see this abbreviated as SOD. It's going to break down those superoxide ions into hydrogen peroxide and oxygen, and then it'll break that hydrogen peroxide down in a secondary step with a different enzyme called catalase, and that'll just break it down into water and oxygen and totally neutralize that reactive compound. And so you'll recognize this word catalase when you get to some of our unknown ID labs. One of them is going to involve catalase. That's actually one of those clinical tests that we do quite readily. And it's a very fast, very informative test because you basically take some of the culture bacteria, add hydrogen peroxide, and if it bubbles, then you know that the catalase was present because that oxygen, literally you can see the bubbling reaction. If there's no bubbles formed, then you know that that bacteria didn't have catalase enzymes. So we also talked about oxygen requirements and how we could distinguish them in the lab. We use thioglycolate media to determine the tolerance. So again, we kind of think of aerobe, anaerobe, can they use it? But it's really also about can they tolerate the presence of it. So we talked about each of these major groups before. Your book also mentions a group called the microaerophiles. It's sort of an in-between stage here. But keep in mind that word facultative. It means they can make do without oxygen. But they technically, if you look at where the heaviest growth is, it's up at the top. And so they prefer oxygen. They can thrive with oxygen, but they can do without, right? Now, some species are more particular about their carbon dioxide requirements, and we would call these capnophiles. In fact, they prefer a higher concentration of carbon dioxide than you would find in the atmosphere. So this allows us to create culture conditions that can isolate certain species. And so when we have diagnostic labs looking for these species, if you just have a higher concentration of carbon dioxide, then it can isolate these particular pathogens. pH. Of course, pH is really going to have a dramatic effect on the shape of proteins. Remember, acids can denature proteins, and so most organisms prefer a neutral environment, so we would call them neutrophiles. Some bacteria and fungi can tolerate the extremes, and a lot of these are going to be those archaea if you're talking about bacteria. So acidophiles would be anything below pH of 6. So keep in mind that things like Helicobacter pylori is the bacteria that causes peptic ulcers. And so we know this species can tolerate the pH of the stomach because they make alkaline byproducts that basically neutralize the acids around them. And those alkaline byproducts are actually from metabolizing urea, which is a nitrogen containing compound. And so we look for H. pylori when a patient can ingest a radio-labeled urea molecule. And if it goes through the stomach and that bacteria is present, they have a urease enzyme that can break that down. And so then we take a sample. They essentially blow into a balloon, and so we take a breath sample that catches some of that that digested urea, essentially. And if the sample that they breathed out has some of that radioactive carbon dioxide as a byproduct, then we know that the bacteria is present. If it's not present in that breath sample, then we know that the organism did not break down that molecule. And so it's a good way to diagnose certain things. Also, some of the species that cause urinary tract infections will have the same type of thing. So it's just all about what kind of enzymes they have and what they can tolerate. Some species like cholera actually doesn't mind the alkaline environment of the ocean. So that's where cholera lives. Now it can also tolerate other conditions like the human body. So it becomes a pathogen if it comes out of that natural range where it would like to be. And even some organisms require a different kind of atmospheric pressure, right? So when we think of barophiles, the prefix baro, right? The weatherman tells you about the barometer readings. And so some organisms require really high pressure around them to survive. And so those are usually going to be archaea that need that really high pressure gradient. But some of them are so highly adapted to that if they weren't exposed to normal atmospheric pressure, they would actually not survive. And so all of this kind of accumulates together to think about what is going to happen in culture. So when we talk about the growth phases in culture, picture what's happening inside of that nutrient broth tube, okay? And at the beginning of an incubation, we inoculate it with a certain number of organisms. Initially, they're going to go through what we call a lag phase. So you should consider 4 things when you're thinking about this growth phase diagram. First of all, what is the population rate or the growth? Is it increasing? Is it decreasing? Or is it stable? What are the concentration of nutrients in that culture dish? Are they absent, increasing, decreasing, or high? What is the enzyme activity in that culture at the time? And it could be either absent, increasing, decreasing, or high. And then look at the concentration of wastes, because there's, this culture dish is going to be accumulating wastes as these organisms start metabolizing things, and it's a closed system. We're not adding anything, we're not taking anything out. So you're really watching sort of what's happening in that culture tube over the course of time. And so the waste concentrations, again, you can think of as either absent, increasing, decreasing, or toxic, because there's a point where too many wastes floating around, you're going to have a toxic environment. So why there's a lag phase, and this is going to vary depending on the organism we're working with, that lag phase, the population, which is really this red line you're seeing, or growth rate, is really stable at this point because nobody's reproducing yet, because they're actually adapting to their new nutrient options, so to speak. So they're in the process of making enzymes so that they can actually use the nutrients that have been provided to them. The thing about living organisms, they're very efficient. They only keep enzymes that they're actually using when they need them. They're going to make them fresh as they need them. Otherwise, they kind of just are wasted. So at some point, once the enzymes are made, they're going to start using the nutrients that have been provided to them very quickly. And we talked about binary fission as a way. We're going to look at some growth curves here in a second, but this log or exponential phase is where you're going to have rapid growth, right? Enzyme activity is going to be very high. Nutrient load is going to start decreasing because it's going to be used. Waste concentration is going to start increasing as well because we're going to be growing new organisms consistently. Now, at some point, those nutrients are going to run out. And so that log phase can't go on forever unless you put them in new media. And so this, what we call the stationary phase, is where your growth rates are stabilized again, and it's not that nobody's growing, it's that the number growing and the number dying have evened out. Okay, so during the log phase, the number growing is much higher than the number dying, so you're going to see an increase. The nutrient levels here probably still decreasing. There's just enough in there to keep a few of them going. The wastes are going to continue to increase because there is metabolism going on. But at some point, not only are the nutrients going to run out completely, but the toxic wastes are going to, or the wastes are going to grow, increase to the point that they're toxic. So anybody that's in there, the number dying is going to outweigh the number that are still trying to reproduce or still hanging on. And so you get what's called the death phase. And so obviously if you're trying to go into that culture tube, depending on the purpose of the culture, if you're going to try to smear that culture and look at it under the microscope, you would want to take a sample somewhere probably right about the middle of that log phase. If you take it too early, your population is going to be so sparse that when you go to stain it, you're not going to have enough cells to be able to look at morphology and arrangement, especially arrangement, because you need that pattern. But if you go too far beyond the log phase, then you're going to be sampling dead cells. And think about how the gram stain worked. If you're trying to stain dead cells, you're not going to retain that stain properly, and you're probably going to decolorize cells that wouldn't have been decolorized otherwise. So it's important to know the growth phase of organisms that you're growing in culture so that you can catch them in the right, kind of catch them in the act at the right time, depending on your purpose. And so keep in mind, bacterial growth is by way of binary fission. They literally, when they get enough nutrients and they have built enough macromolecules in their cell that when they get basically twice as big for their britches, they just split down the middle and start over, right? And so there's a wide range of doubling time or generation time in bacteria. But, some of them are as quick as 10 minutes. Others are going to take about, up to 30 days, and that'd be something like tuberculosis. It's really hard to culture tuberculosis organisms because they're very slow to go through this process, partly because their cell wall has that waxy component, and that's really expensive, nutritionally speaking. But on average, it's somewhere between 30 and 60 minutes, and so you can see why it's really critical for food handling safety, right? If you have leftovers sitting out on the table that have been contaminated and it's got one of these species that can grow in 30 minutes or less, by the time you put that in the refrigerator, it's already got a heavy load of bacteria growing on it. So just understand that the process here is you're just doubling the population every 30 minutes or so. And so if we were to graph this, you would see a dramatic difference between an organism like a yeast, right? You would have one yeast putting off one little sort of mini-me yeast at a time. Budding would be something that you could graph arithmetically, right? Same thing with something like humans. Now, yes, we can have more than one baby at a time, but the generation time is usually one by one over the course of time. Bacteria, on the other hand, because they double their population with each reproduction, that binary fission means that if you were to put them on the same graph, right, you're going to run out of graph very quickly. You're going to need a logarithmic graph. You would need this axis over here where it says number of cells. You would need that to go by, 1 to 10 to 100 to 1,000 for that graph to accommodate that growth rate. And so this is the difference between what we call linear and exponential growth. So we call it symbiotic if they live together. How they live is going to be usually categorized in one of these types of relationships. And so usually the organism in column 1 here, organism 1 would be the microorganism. The organism in column number 2 would be the host. So if we're looking at human body and the organisms that live in and on our bodies, we would be organism 2. And so many of the organisms in our body are what we call mutualistic relationships. So mutualism is where both parties benefit, right? So look at the example here, right? The bacteria in your colon. So the E. coli that lives in your colon. There's good E. coli and there's bad E. coli, but the type that typically lives there benefit from being there, right? What's their benefit? Well, they have a nice, warm, moist place to live with plenty of nutrients. What's the benefit to the host or ourselves? You may recall from A and P, the bacteria in your colon actually provide about 50% of the vitamin K that you need. Without them, we would have bleeding, right? Because vitamin K is really critical for clotting factor formation. So that's one of those long-term side effects of antibiotics is bleeding sometimes. Because if you kill off the good guys, you don't have enough vitamin K in your diet to make up for the fact that they usually provide about half of what you need. In some cases, and actually most organisms on the human body are what we call commensal, meaning that the organism is benefiting by having a place to live and plenty to eat, but they're not going to cause any harm to the human, and they're really not providing any benefits. So mites, for example, they really don't cause any problems for the human host. Some people would put Staphylococcus in this category, or at least in somewhere between mutualism and commensalism. And I mean the good Staphylococcus, right? We have good and we have bad Staphylococcus. What they get out of it is, again, a home and a plentiful nutrition, but what we get out of it is really competition. They're there to keep the bad bacteria and the bad yeast and other organisms from growing on our skin. So Staphylococcus would probably be a mutualistic relationship. Amensalism is where really the host is not necessarily harmed or benefit, but the organism that lives near or on is being harmed. So, for example, the fungus secreting an antibiotic creates a harsh environment for the bacteria that live not nearby. Now, again, some scientists would say that antibiotics are actually providing competition or reducing competition by killing off the neighbors. So that would actually give them a benefit. So again, there's a little bit of gray area when it comes to these terms in certain relationships. But where we're going to see the most diseases, it falls under this parasitism relationship, right? So this is where the organism, the microorganism, benefits, but the human host is harmed, right? So for example, tuberculosis, obviously, if it has progressed to disease form, then that would be parasitism, right? So usually when we're talking about tuberculosis, again, many people are walking around with the bacteria in their lung, but they're not actually being harmed. So in that case, you might think of it as a commensal relationship. But if you actually progress to active tuberculosis, it would clearly be a parasitism relationship. Parasites would also include things like ticks and mosquitoes. That's usually where people think of the large word parasitic, but really it's all about what the relationship between the two organisms is about. Now, not all organisms are symbiotic, but they sometimes have an effect on each other. So if it is a synergistic relationship, very oftentimes this is where biofilms come into play. So biofilms very oftentimes involve mixed infections. There are a lot of different species present. They may or may not influence each other, but if they do, we're looking at situations, especially in the human body, things like gum disease, right? There are certain bacteria, if they're present, that they can cause biofilms to form, and then that just creates a really persistent problem where your immune system can't get to them. Even proper brushing and flossing can't get to them, so it becomes a problem and creates long-term disease, periodontal and gum disease. Urinary tract infections that continuously come back, so people that have chronic or persistent urinary tract or pneumonia infections, very oftentimes it's because there's a biofilm form. So you may have antibiotic treatments that are ineffective, things like that. Especially things like pneumonia. We now know that cystic fibrosis patients end up with biofilm formation because mucus is not produced properly. And so when they get these chronic pneumonia infections, it's usually because the biofilm is in there and it's hard to get rid of. Antagonistic relationships would be those where the members that are that are sharing an environment necessarily are competing with each other. So we mentioned how E. coli is good competition in the gut. If you take away that good bacteria, not only will you not get enough vitamin K, but you will also allow things like C. diff to take over. So competition is really a good thing. It might sound like antagonism is not a good thing, but it's actually one of those things we count on in and on the human body. So that competition in our body would be things like the gut flora or those staphylococcus on your skin. They actually are providing some competition for the bad guys, against the bad guys. Microorganisms that are producing antibiotics are sometimes referred to as antagonistic, right? They are competing with their neighbors. So by producing an antibiotic that kills their neighbor, they reduce the competition. They're not going to fight for nutrients or space if they kill off their neighbors. And so biofilms, again, we've met them several times already this semester. We're going to continue seeing them over and over. So really the basic steps of forming a biofilm involve at least some type of pioneer bacteria, right? And these would be the guys that have to have a fimbriae or a capsule or something that allows them to stick and colonize the surface. Then they start to secrete something that becomes the material that other organisms can stick to, right? And so by providing these attachment points for other species, then it just sort of contributes to the layering of the biofilm. And so once you have a certain number of organisms present, that quorum sensing is going to kick in. And that's where they're going to start releasing chemicals to either communicate with their neighbors or even invite other species. Once they're there, and again, not all biofilms involve multiple kinds of species, but once if they do, that quorum sensing doesn't even require communication within species. Sometimes it's crossing over between species, and that usually requires that conjugation, right, where they're communicating through pilus and they're sending DNA and they're actually triggering each other to make other things like toxins or other chemicals that are just going to recruit more types of bacteria. And so once the biofilm is formed, it's almost like an entity of its own, and it becomes a source. And this is where those persistent infections become a problem, right? You can kill off the top layer maybe with antibiotics, but once the antibiotics are gone from the body, if you haven't taken the full course, or if even the full course of antibiotics may not be enough to get rid of the entire biofilm, and so it just becomes a source of infection. And so this is where those bacteria then, when they leave the biofilm, they can go set up camp somewhere else and just start all over. So clearly biofilms are a big problem. We're going to talk a lot about them again when we get to antibiotics and again as a virulence factor as we go through the semester. So that's it.